Dear students, you are welcome in this class. Today we are going to have a practical on total nitrogen estimation in soil as well as in plant sample. Soil, you know that we need to know status of nitrogen in soil. If we can know the status of nitrogen in soil, that is total nitrogen, it can tell us, it can give us indication that how much is the potential of the soil to supply nitrogen. In general, higher being the total nitrogen, we can expect more mineralization of nitrogen and we can expect more supply of nitrogen. So why do we estimate total nitrogen in soil? To check the availability of nitrogen during the growing season. How much of it can be made available for the plants? And why do we estimate total nitrogen in plant samples? So in plant samples, anybody can tell me what is the purpose of estimating nitrogen in plant samples? Why do we estimate nitrogen in plant samples? Anybody can come. Sneha, why do we estimate total nitrogen in plants? Yes.
So that we can estimate the uptake of nitrogen by the plants, the accumulation of nitrogen in the plant.
Okay. Chandrakant, why do we estimate?
Yes.
What is the purpose? Why we do it?
Sir, for fertilizer application, how much we apply fertilizer?
Okay. So he is giving indirect answer. Indirect answer to apply nutrients, to apply nitrogen. We estimate the nitrogen in plant sample. Fundamental purpose during the growing plants. During the growing plants, we like to see the adequacy of the nitrogen. Whether the plant is having sufficient levels of nitrogen or not. This plant testing for nitrogen is basically to see whether it is having enough nitrogen in it or not. If it is having low levels of nitrogen, below the critical level, then we can apply some nitrogen from outside. This is one purpose of knowing the nitrogen in plants. Second is to know the uptake of nutrients, uptake of nitrogen. We can easily calculate the total nitrogen removed by plants. Plants, if we estimate the concentration. So how this concentration will be useful in calculating the uptake of nitrogen or any other nutrient? Okay, we have estimated concentration in straw, concentration in grain. Then how it will be useful to know the uptake of the nutrient or uptake of the nitrogen? Very simple question. Yeah, Shubhjit.
Yes, sir.
If we know the concentration of nitrogen in the plant tissues at a particular stage, and we want to compute or calculate the uptake of nitrogen, then how can we do it? How can we calculate the uptake? If we know the concentration of nitrogen in plant tissues.
So uptake is calculated by multiplying the concentration with dry matter.
Yeah, fantastic. So we need to know dry matter also, dry matter yield simultaneously, depending upon the stage of the crop. Suppose you have cereal plants like maize, wheat, rice, etc. If they are in early stage, that is vegetative stage, when there is no reproductive stage, at that time you can harvest the whole plant material and know the dry matter of this plant material and multiply it by concentration of nitrogen, you can get the uptake. But when you have grain developed, grain formation has happened, you have grain as well as straw or stover, then you can separately estimate the concentration of nitrogen in stover or straw and grain. That will help us to calculate the uptake or removal of nitrogen by grain as well as straw. Then you can total it and calculate that this much is the nitrogen uptake kg per hectare in the plant. So knowing the nitrogen concentration, total nitrogen in plant, concentration, we can calculate the uptake or removal. And secondly, we can also calculate the protein percentage. If I ask you, calculate the protein percentage in wheat grain, then you need to know only nitrogen. You need not to analyze proteins directly. You can just analyze the total nitrogen and multiply it with a factor, and then you can get the crude protein content in the plant material, in the seed, isn't it? So also to compute, to know the proteins, we need to calculate total nitrogen in the plants. Here, students, there is no available nitrogen kind of things in the plant sample. Here, only one thing is, that is total nitrogen in plants. However, in soil, you have total nitrogen and available nitrogen. Can somebody tell me difference in total nitrogen in soil and available nitrogen in soil? What is the difference in total nitrogen in soil and available nitrogen in soil? I discussed at some other point also.
So total nitrogen would include organic and inorganic form both. But
Okay.
when we talk about available nitrogen, then mostly the plants will take the inorganic form only.
Okay. So she's saying that organic nitrogen means, total nitrogen means organic plus inorganic. However, available nitrogen is inorganic nitrogen. She is partly true, because inorganic nitrogen, available nitrogen also includes readily oxidizable nitrogen from organic matter. Organic matters, on an average, supplies about 2% of the total nitrogen to the soil, that is mineralized, or 2% nitrogen present in organic matter. Whatever nitrogen is present in organic matter, 2% can be mineralized into inorganic forms during the cropping season or during 1 year. In 1 year, whatever total nitrogen is there, suppose it is 2,000 or say 3,000 kilograms per hectare. 3,000 kilograms per hectare is the total nitrogen. So out of this, 2% will be made available to the plant during the growing periods. So 2% of 3,000 means 60 kg. So 60 kg nitrogen will come from organic materials, which is called as mineralizable nitrogen, or potentially mineralizable nitrogen, or readily oxidizable nitrogen. There are three different names: potentially mineralizable nitrogen, readily oxidizable nitrogen, and so on. So if we know the contribution of nitrogen that will come from organic sources, then it will help us to reduce the amount of fertilizer, reduce the amount of fertilizer. So therefore, available nitrogen in soil means inorganic nitrogen or mineral nitrogen, which is ammonical and nitrate nitrogen, plus readily oxidizable nitrogen. That is your available nitrogen, which is part of the total nitrogen. Now we come to the objectives of this exercise: to determine total nitrogen in soil and plant samples, that is in grain and straw. The role of nitrogen in plant will be known today, and relationship between total Zeldal nitrogen and crude protein content in different plant tissues, and certain numericals. Now question comes, why nitrogen is important for the plants? What is the role of nitrogen in plant? Tell me quickly. Yeah, Chandrakant. What is role of nitrogen?
For making and so many
There are so many ways.
protein enzymes and and like
Achappa, I think his voice is not clear actually. He may be right. Achappa,
Sir, it is helpful in protein synthesis.
yeah, it is part of the proteins necessary for protein synthesis, for chlorophyll synthesis, formation of, it is constituent of your DNA, RNA, and it gives greenness to the plant because it is part of the chlorophyll. So it has very important role. If you see from outside, in cereal crops, it has very profound influence on tilting of the crops, growth of the crops, leaf area index. It can increase leaf area index. It can increase number of leaves, or you can say it can increase or improve vegetative growth of the plant. And it also helps in the root growth. Nitrogen is also like phosphorus. Nitrogen is also necessary for root growth. So it is the most important nutrient in crop production, this nitrogen. Now you see it is just a repetition what I already discussed. So total nitrogen in soil, you can see organic nitrogen, ammonical nitrogen, nitrite nitrogen, and nitrate nitrogen, mineral or inorganic nitrogen. This ammonical, nitrite, and nitrate nitrogen makes up your mineral or inorganic form of nitrogen. But this can be supplemented. This can be supplemented. This pool, mineral pool or inorganic nitrogen pool, can be supplemented through fertilizer. You can add urea to increase this mineral nitrogen or inorganic nitrogen in soil. You can add organic matter to the soil or manures to the soil, and after decomposition of those organic materials, they can supply mineral nitrogen. And you know what are the steps through which the organic materials are decomposed to produce nitrogen, which is called as mineralization. So organic forms of nitrogen will also give you inorganic form of nitrogen. At any given time, 95 to 99% of nitrogen in soil is in organic forms. Please do remember, see 95 to 99% means very high proportion of nitrogen in soil is in organic form. That means remaining 1 to 5% is in inorganic form. Inorganic form is 1 to 5%. And mineral nitrogen in soil accounts for, on an average, 2% of the total nitrogen in soil. So this is actually mean value. This is actually mean value. The range can be from 1 to 5%. Is that clear? Now, what are the organic forms of nitrogen in soil? What are the organic nitrogen compounds in soil? So organic soil nitrogen occurs as proteins when it is undigested. When you add fresh organic materials to the soil or manures to the soil, nitrogen may be mostly in proteins, amino acids, amino sugars, and other complex nitrogenous compounds. The proportion of soil organic nitrogen in these various fractions means where actually the nitrogen is there in organic compounds. So you can see most of the nitrogen will be in amino acids. 30 to 45% is in amino acids, and bound ammonical form is 20 to 35%. Acid insoluble is 20 to 35%. Amino sugars 5 to 10%. Unidentified is 10 to 20%. So these are the compounds, nitrogenous compounds in soil. Now see, most forms of organic nitrogen cannot be taken up by plants. Underline this statement. Most forms of organic nitrogen cannot be taken up by plants with the exception of some small organic molecules. That means organic nitrogen can also be absorbed by the plants. Students always think that it is mostly inorganic nitrogen, which is always taken by the plants. But many times, plant can also remove dissolved organic nitrogen or soluble organic nitrogen, which is in form of amino acids and amino sugars, and maybe some unidentified molecules that can be taken up by plant in organic forms. And you see the organic nitrogen and inorganic nitrogen is in equilibrium, depending upon soil and climatic conditions and also management. So organic nitrogen can be mineralized to inorganic form. You know the process of mineralization of organic nitrogen or process of mineralization of proteins. How proteins mineralizes means how proteins are converted into nitrate. You can see inorganic form. Can anybody tell me how organic nitrogen with the help of microbes is converted into inorganic forms? Who will tell me? Come forward, Shubham. Shubham,
Yes, sir.
releases ammonium. Later, this ammonium converted through nitrification process into nitrate.
start from beginning. How organic nitrogen is converted into amino acids, and then how amino acids are converted into urea, and how urea is converted into ammonical form, and then to nitrate forms. Complete steps.
The first amine
Yes. Our voice is not clear. Other person.
ionization will occur
आपकी
through microbes.
okay.
It is followed by ammonification.
Okay. Your voice is coming broken. Anyways, I think,
Sir,
yeah.
Sir, first aminization will occur. It is followed by ammonification. It is followed by nitrification.
Okay. So what are the compounds that are formed under these steps?
Sir, in aminization, organic matter converted into amino acid. Through ammonification, it will converted into ammonium ion. Later, through nitrification, it get converted into nitrate.
Okay. So I was telling you that inorganic forms of nitrogen are ammonium, nitrate, and nitrite. So presence of nitrite is not there in the soil. When nitrites are formed, immediately within seconds, they are converted in the form of nitrates. So whenever you analyze nitrite in soil, you will not find any quantity of nitrite in the soil. And nature has done it rightly so. Because if nitrites are there in the soil, they are very, very poisonous to organisms producing it, number one. And second thing, they are poisonous to the plants also. So nitrites are very, very ephemeral, very, very temporary. So whenever you calculate mineral nitrogen or inorganic nitrogen, it is always ammonical and nitrate nitrogen. Nitrite nitrogen is generally not included. So what steps she told are right steps, and that is the mineralization. And mineralization means aminization, ammonification, and nitrification. Now you see what happens in aminization as discussed by her. So proteins, you can see aminization, what happens. Proteins in the presence of water is in soil, bacteria and fungi are in soil. And as you know, the bacteria dominate in neutral to slightly alkaline soil, and fungi dominate in acidic soils. So if you have acidic soils, then decomposition of proteins will be dominated by fungi. If you have neutral to alkaline soil, decomposition will be dominated by bacteria, and they are heterotrophic organisms. Heterotrophic means they depend upon for food and energy on other organism or organic materials. Then these proteins are converted into amino acids. You can see amino acids. Amino acids, you have R, C, COH, NH2H. So this is general formula of amino acids. And then this is NH2. NH2 is formula for amines. amines. So while protein is decomposed or mineralized in aminization, three different products are formed. You can see three different products are formed. One is amino acid, other is amines. Amines is RNS2, and other is urea. So three things are produced when the proteins are aminized. Remember name of these products. What are the products of aminization? Amino acids, amines, and urea. You can see the formula of urea, NH2CONH2 plus CO2 plus energy is released. And you see amines are RNS2 and amino acids are RNS2COH and H. What is this R? What is this R?
So any alkyl group.
Pardon?
So any alkyl group.
Okay, you explain to your friends. Please explain more to your friends if you have idea.
So it could be CH3 or C2H5 or anything.
Good. So what message here is, why I am asking it, so that little bit of nomenclature of organic compounds, you should learn what is meant by eth, और 3 को क्या कहते हैं? 4 को ब्यूट कहते हैं, ब्यूटाइल, ब्यूट. आप देखना उसको, you see it. So R is your certain groups, alkyl group or what she said. Now see, R refers to alkyl, alkenyl, aryl or other groups. This R. Now see ammonification, amines. This is shown for amines. So getting water, microbes are there, and this is converted into ammonia. And then water, it is hydrolyzed and forming ammonical ions and hydroxyl ions. So you can see in ammonification, amines have been converted in the presence of organisms, heterotrophic bacteria, fungi, nematodes. It is converted into ammonia. And this ammonia is, after getting water, it is dissociated into ammonium plus hydroxyl ions. And ROH is left and plus energy. So this is, the process is ammonification. Here ammonium is formed. Now depending upon the pH, this ammonia is very, very unstable. Either it will make ammonium or it will be lost. Depending upon the soil pH, if soil pH is high, it will be released as ammonia. So volatilization will be there. Now you see the fate of ammonium. Right now you have seen this ammonium. Ammonium was made through ammonification. You see this ammonium. And what will happen to this ammonium? The ammonium produced here through ammonification can follow different routes or different fates. First thing is that some of the ammonium produced here by nitrification can be converted to nitrite and nitrate. So some will be channeled to nitrification. Some may be absorbed by higher plants, particularly aquatic plants like rice. They can absorb nitrogen in ammonical forms. It can be utilized by microbes also. Heterotrophic microbe like bacteria can utilize it for their function. That is decomposition of residues. And this will be called as immobilization. So some of the nitrate ammonium can be immobilized by microbes which are involved in decomposition. Some of the ammonium can be fixed as biological unavailable nitrogen in the lattice of certain clay minerals, which is known as ammonium fixation. So ammonium fixation can also happen in the lattice of the clays. In between the layers of the clays, you have some interspaces. In those interspaces, this ammonium can enter, which is known as ammonium fixation. Did you know that ammonium fixation? It is different from nitrogen fixation. So what is the difference in nitrogen fixation, ammonium fixation, and phosphorus fixation? These three terms are there: nitrogen fixation, ammonium fixation, and phosphorus or phosphate fixation. What is the difference in three? Mohdeh? Ammonium fixation, nitrogen fixation, and phosphorus fixation. What is the difference between these three? Mahoday, are you there? Hello? Mahoday? Amandeep?
Yes, sir.
Tell me the difference among the three.
Sir, ammonium fixation is the adsorption of ammonium ions into soil layers.
Clay layers. Okay.
Yes, sir.
And nitrogen fixation?
Nitrogen fixation is the conversion of free nitrogen gas into nitrate form, sir, and then absorbed by plant roots.
I think you are not fully correct. And phosphorus fixation?
इसका नहीं पता सर.
कोई बात नहीं. Any other Kanchan? Kanchan? Rimjim?
Yes, sir. Sir,
What is-
phosphorus fixations when like in acid soils, if the iron and aluminum is in high quantity, then phosphorus is fixed in unavailable form. And in alkaline soil, it is fixed by calcium on the calcium carbonate, so it becomes unavailable.
Okay, this is phosphorus fixation. Then nitrogen and ammonium fixation.
Sir, ammonium fixation is the in clay minerals on the negative sides of the clay mineral. And
Yeah.
sir, nitrogen fixation is the sir conversion of the atmospheric nitrogen into ammonia.
Fine. My purpose is that you should know these simple terminologies, and these are very, very important. Now I make it clear to all of you. First, let us see your ammonium fixation. So what happens, certain point of time, you have sufficient quantities of ammonium in the soil. Either it is coming from fertilizer, or it can come from organic sources. So what happens, this ammonium has almost identical size. The size of this ammonium ion is almost identical to the size of the spaces in clay micelle or clay minerals. Inside, they have spaces where this ammonium can enter, and it is temporarily fixed. It is temporarily fixed in the clays, which is known as ammonium fixation. It has very, very high significance in potassium nutrition also. So because potassium ions and ammonium ions have similar ionic radii, their radius is similar. Therefore, both potassium and ammonium can enter the clay micelle, because of which the potassium and ammonium both can go for fixation. So you can remember potassium fixation means entry of potassium ions into clay micelle or its interspaces in the clay minerals. Potassium can enter, then potassium fixation, ammonium enters, then ammonium fixation. So many times what happens, your soils may be having sufficient nitrogen which has been fixed in the ammonium in the form of fixed as ammonium fixation. So what you need to do, in that case, you need to apply potassic fertilizer. To increase the supply of nitrogen, you need to add potassic fertilizer that will release ammonium that has been fixed by the clay minerals. You understood? And vice versa. Sometimes there may be sufficient potassium, but it has been fixed by the clay minerals in their spaces. Then to take it out, you need to add some ammonium also. If you add ammonium, this potassium will come out from the clay minerals. And nitrogen fixation, you know, that is biological nitrogen fixation, which is fixed by organism, symbiotic and non-symbiotic. So these are the differences. So now you have seen that how this ammonium, fate of ammonium, it can go to nitrification, it can be absorbed by plants, it can be used by organisms, it can be fixed as ammonium fixation, and it can be converted into ammonia. So there are variety of routes these ammonium can go. And you can add one more. That is, I told you, ammonium leaching. So ammonium can be leached from the soil also, particularly in soils where you have very less cation exchange capacity. And one thing Rimjim has incorrectly told, no problem, we make mistakes and it is time to correct our mistakes. So that is actually, she was saying that it is absorbed on the clay minerals. It is not absorbed, it actually enters into the spaces. That is the mistake, and we can correct it, no problem. And this is about the roots of ammonium. Now, coming to the next stage, that is immediately after ammonification, now you get nitrification. So immediately after ammonification, that is conversion of organic nitrogen to ammonia, then hydrolysis, conversion in water of ammonia to ammonium is followed. I have already told you this ammonia is released, you see amines, water and some organism, ammonia is produced and energy is also produced. And then this is hydrolyzed, means it reacts with water and it forms ammonium plus hydroxyl ions. And this ammonium fate, you have seen what is going to happen to this. Now, ammonium and now what happens, the relationship between ammonium and ammonia. Ammonium and ammonia. Right now you have seen that ammonium ions are formed after hydrolysis. So ammonium and ammonia, nitrogen are closely related. Ammonia is a gas and at high pH levels, favors this form. This form is formed at high pH and it can quickly escape from the soil into the air. Ammonium is a soluble ion and favored by low or moderate pH. So if your pH is lower or moderate, then this ammonia will not escape. This will remain in the form of ammonia. Ammonium is attracted to negatively charged soil particles held as an exchangeable cation in the soil. So it is attached. And what is double diffused layer? What is double diffused layer? Can anyone tell me what is double diffused layer? Who is from soil science? Who is from soil science? Is there any student from soil science? Kanchan? Apurva, are you from soil science?
No, sir.
Who will answer it? I expect answer from soil science student. However, anybody can answer it.
So it is a double. So it is a double layer in which the negative charge of the clay, the positive charged ions are held, and after the positive charge, some more negative ions are held on the positive charge, and it makes the Helmholtz double layer, which are weakly held. The outside ions are weakly held.
Fantastic. Fantastic. Where you read it? Where you read it? What was the reference to study this point?
So ISA.
ISA, Indian Society Agronomy book.
So
Any-
Indian Society of Soil Science.
Okay.
ISSS.
Fantastic. Your answer is correct and you are absolutely right. And for more, students are advised to see what difference she is telling and other sources, nature and properties of soil. In Pradeep's book, you can find this double diffuse layer. Ammonium is attracted to negatively charged soil particles held as an exchangeable cation in soil. So it can be absorbed by the colloids, soil colloids, whether it is inorganic colloids or is it organic colloids. Ammonium is a common soil nitrogen form. This is also different, and ammonia is not, because ammonia is not going to stay in soil. Either it will be lost or it will be converted into ammonia. there are only two ways for ammonia. Either it will escape or it will convert to ammonium form. Ammonium can also be trapped between the layers of the clay minerals, which is known as ammonium fixation. This is discussed. Now, coming to the available nitrogen, as already discussed, it is mineral nitrogen, which is ammonical and nitrate nitrogen, and potentially mineralizable nitrogen in organic matter, or readily oxidizable nitrogen that can be readily oxidized. So these two make up the available nitrogen, means the nitrogen which can be utilized by the plant or which can be part of the uptake. Whatever uptake is there by the plants will be in these two forms or will be from these two sources. One is mineral nitrogen, ammonical and nitrate, which is freely available in soil, and other potential source is organic matter that can be mineralized during the growing period of the crop, which is known as potentially mineralizable or readily oxidizable nitrogen. Remember this, in general, 2% of organic nitrogen in soil is converted to mineral forms in a year. This is average value, general value. It may vary from, say, 1.5 to 3.5%. Somebody can say 1 to 4% also. So this value you find in literature somewhat changes with the people. But what I am showing you here are very general range that you can remember. In general, 2% of organic nitrogen in soil is converted to mineral forms in a year. Range being 1.5 to 3.5%. Most nitrogen release from organic matter occurs during growing season, providing a continuous supply. If your nitrates and ammonium are exhausted, these nitrate and ammonium will remain present in the soil solution. They are mostly 99% of ammonium and nitrate will be there in the soil solution. Of course, some of the I take back my statement. Some of the mineral nitrogen will be in soil solution, particularly nitrate. 100% nitrate will be in soil solution, and some ammonium will be there in the soil solution, and most of the ammonium will be as exchangeable ammonia. It will be absorbed at colloids, inorganic colloids or organic colloids. Is it clear to you? Because we have high proportion of ammonium nitrogen as exchangeable ammonium. Small fraction is in solution forms. Most nitrogen released from organic matter occurs during growing season, providing a continuous supply. So after the decomposition of organic matter, some of this will go to the soil solution and some will go to the exchangeable sites. So therefore, it will be in equilibrium. This is because the microbes responsible for releasing nitrogen from soil organic matter require some soil moisture also. Now see this picture, very interesting picture about,
Sir?
yes.
Sir, please explain the last line in the last slide.
This is because the microorganism responsible for releasing nitrogen from soil requires some, this is not soil moisture. This is soil nitrogen. This may be soil nitrogen. This is not soil moisture. This may be mineral nitrogen, either nitrate or ammonia.
Okay, sir.
Yeah, thank you for correcting me. Thank you very much. Now you see this picture by Carson and Phillips. It is very simple to understand. Just come to the nitrogen fertilizer. You have added some fertilizer, and you know fertilizer, nitrogen fertilizer mostly means urea, or you can say ammonium sulfate or some other fertilizer, and it will become part of the nitrogen pool, part of the nitrate and ammonium pool, which is mineral nitrogen. So initially it will become part of the mineral nitrogen. Under normal conditions, in how many days urea can provide nitrate? in how many days urea will convert into nitrate under normal moisture, normal temperature, normal soil conditions? Any answer? Suppose I put urea today in my field. I have grown, say, rice crop. After how many days nitrate will be made available to the plants? Normal urea. Any student? Mohoday? Mohoday? I think he just connected to the Zoom link and he left it. So, any answer, Apurva?
Sir,
No,
after
sir.
8 to 10 days.
After?
8 to 10 days.
Right, correct. So it may be 8 to 10 days or 7 to 10 days, something like this, under normal conditions. So here you see nitrate and ammonium nitrogen, that is mineral nitrogen, 2% nitrogen in soil, taking the average as 2%, you can see. And some of the mineral nitrogen which is present in nitrate or ammonium form, here it is shown nitrate leaching, but it can be ammonium leaching also. And you can see some of this can be taken up by the plants. Some of this can be taken up by the plants. And you can see that the roots or leaves, they can become part of the soil, and then they can become part of the organic nitrogen, which is 98% in soil. You can see range is from 95 to 99%. Here it is shown 98%. So, and then it can mineralize. It can mineralize and it will also supply to the pool of mineral nitrogen. So this picture conveys the message that mineral nitrogen can come from fertilizers and it can also come from organic nitrogen after the mineralization. And it can be reverse also. This mineral nitrogen can be immobilized into organic nitrogen by soil microbes. And a small portion of organic nitrogen can be taken up by the plants. It is shown here. Some of in the dotted line, you see arrow, dotted line. Here, some of the organic nitrogen can be taken up by plants directly. Healthy plant foliage generally contains 2 to 4% nitrogen, depending on plant species and age of the leaves. So here you can see healthy plant foliage contains on an average 2 to 4% nitrogen. Nitrogen, the role of nitrogen already discussed. It is part of essential compounds like amino acids, the building blocks of proteins, including enzymes, nucleic acid, and chlorophyll. A good supply stimulates root growth and development, as well as uptake of other nutrients. So nitrogen has many important roles. Now coming to the main method, Geldal method. So what is the basic principle? Basic principle is that organic nitrogen in sample is converted to ammonium nitrogen by digestion. Be careful now. Organic nitrogen in sample. You have a soil sample or plant sample. Whatever sample you have, majority of the nitrogen in both is in organic form. So organic nitrogen in sample is converted into ammonium form. So finally, you need to convert all kind of nitrogen into ammonium form by digestion with the concentrated sulfuric acid containing potassium sulfate or sodium sulfate that promote this conversion. That is the basic principle for digestion. Because nitrogen estimation has three parts. One is digestion, other is distillation, and third one is titration. So in digestion, you decompose the organic matter, you mineralize it artificially with the help of sulfuric acid. Sulfuric acid has characteristics to burn the organic material. It releases carbon dioxide and water. And then the elements may come in inorganic form. Here we are dealing with nitrogen. It will be converted into ammonium form. And now we go for the second step, which is distillation. So whatever ammonium we have got, we want to convert it into ammonia so that this released ammonia can be collected in boric acid. And then we can estimate or we can titrate the present nitrogen, which is there ammonia in the boric acid. So ammonium nitrogen is determined from the amount of ammonia liberated by distillation of the digest with an alkali, that is NaOH, sodium hydroxide. It will volatilize the ammonia from ammonium sulfate or ammonium. This is your distillation. And then this ammonium will be collected in beaker containing boric acid mixed indicator. So then the color, when the ammonia enters, ammonia is alkaline in nature, when it enters the boric acid mixed indicator solution, the bluish green color appears. The new color will be bluish green. Then we go for titration. You can have 0.1 normal sulfuric acid in the burette, in the burette, and do the titration with the boric acid containing mixed indicator. And then you can note down the reading of the sulfuric acid. And at the end point will be again pink or red color will reappear at the end point. So these are three basic principles to make you understand more, to have more clarity. I repeat again so that each one of you is able to understand three different steps. So friends, here come to first step, that is digestion. You have taken a soil or plant sample. You have put in the Geldal flask. This is burner or any heating. It can be heater, it can be burner or any source of heat. And then in this particular Geldal flask, you need to add soil sample or plant sample. If you are adding soil sample, if you are using soil sample, then you need to add two more compounds here. Besides sulfuric acid, you need to add sulfuric acid to decompose organic matter. But you also need to add here salicylic acid and sodium thiosulfate. Salicylic acid and sodium thiosulfate here, if it is soil sample. Because in soil sample, you have three kinds of nitrogen. One is organic nitrogen that can be decomposed by sulfuric acid. Another is inorganic nitrogen. Inorganic nitrogen is ammonium. Ammonium can easily form ammonium sulfate because you have added sulfuric acid here. So ammonium can be easily converted into ammonium sulfate. Organic nitrogen can be broken down by sulfuric acid that can be converted into ammonium sulfate, because decomposition of organic matter will result in ammonium form of nitrogen, and that ammonium can react with sulfuric acid to form ammonium sulfate. The third form is nitrate. So if you want to have nitrate in your total nitrogen, you will also try to include nitrate, and to make this nitrate, to convert this nitrate into ammonium form, you need salicylic acid, and this sodium thiosulfate. These two chemicals are required to reduce nitrate present in the soil sample into ammonium form. And once it has come to ammonium form, then it can be directly converted into ammonium sulfate. So after digestion of soil sample, your all kind of nitrogen, whether it was organic nitrogen, whether it was ammonium nitrate, ammonium nitrogen, or nitrate nitrogen, it will be converted into ammonium sulfate. Means all the nitrogen, total nitrogen has been converted into ammonical form in this digestion process. So here you have used salicylic acid and sodium thiosulfate. Two extra chemicals have been used. But if it is plant sample, you need to add only sulfuric acid. You need not to add salicylic acid and sodium thiosulfate. In both the cases, whether it is plant sample or animals, plant sample or your soil sample, you need to add digestion accelerator or some mixture is added to raise the boiling point. For this, you can add sodium sulfate or potassium sulfate and copper sulfate. So I will show you how much is the proportion of copper sulfate and sodium sulfate. Under both the cases, whether it is for plant or for soil, they raise the boiling point, means if you use them, less energy will be consumed and the process of decomposition will be accelerated, it will be fast. So this is how we digest the soil or plant sample for total nitrogen, where finally you have got all kind of nitrogen in ammonical form. Is there anybody who could not follow it, who could not understand it? Kunal, is it clear to you? This
Sir,
digestion clear?
yes, sir.
Any doubt?
No, sir.
Okay. Now the digestion is complete, and to know that this has been completely digested, the contents in the Jaldal flask should be clear, should be very clear. There should not be any suspended particles, and this is very light blue in color because of addition of copper sulfate. We also add copper sulfate in this. Therefore, this liquid in this looks like light bluish in color. And now we can straightway take this Jaldal flask, put some water into it. Immediately we can put some water into it, say 150 ml, to cool it down. Because if it is too hot, then we can lose some ammonia. So we add about 150 ml of water and keep it for a while, say for 30 minutes, we can keep it to get it cooled down. And once it is cooled down, we can take it to the state where this is also Jaldal flask. You can see the shape is a bit different, but it is also Jaldal flask. So you can take it to the distillation unit. For distillation, number of options are available. Some are locally made distillation assemblies where you can do 5 samples, 6 samples, or 8 samples at a time. And there are certain assemblies where you get option of 1 sample, and some ready-made equipments are also available. Automatic equipments are available, which can do all the 2 processes together and also digestion as well. So those are automatic total nitrogen analyzers, you can say. And then in those, you can just put the sample for digestion, and then immediately you can go for distillation and then titration. But under most of the Indian laboratory conditions, we do not have those expensive equipments. So people work with these kind of assemblies. So now you are going to proceed to distillation. Here distillation means this ammonia will be liberated in the presence of a strong alkali, that is sodium hydroxide. So you have Jaldal flask. Here your digested material is there. You have added some amount of water, distilled water in it, and it was cooled down. And then you have added some ammonium hydroxide. 40% ammonium hydroxide, say 120 ml of this is added here. I will check whether it is 120 ml or 150 ml is added. Then the ammonia will be liberated. Ammonia will be liberated. When you start heating it, then ammonia will be liberated, because the sodium hydroxide will react with ammonium sulfate. It will react with ammonium sulfate and ammonia will be released. And along with this ammonia, some water vapor will also go. So, and then they will pass through the tubes or these channels. And here you have condenser to cool it down, to cool down the ammonia, which was very hot at this stage. It was in gas form. And here the water is circulated around this tube. So this is known as condenser to cool it down. And here there is input of water, and then it will cool down the tube and the materials, and then the hot water will be out from this outlet. So this ammonia will be collected in the boric acid. Here you have 4% boric acid mixed indicator solution, which normally is 20 ml. So this I will tell you how to make the boric acid mixed indicator solution, means boric acid plus indicator. This is pinkish in color, red or pinkish in color. And when ammonia starts coming to this, it will convert into bluish green. It will convert into bluish green. The red color will disappear. It will convert into bluish green. So this is how your ammonia will be distilled. Now question comes, when should I stop this distillation? When should I stop this distillation? When I may consider then whatever ammonia was present in the sample has evaporated, has been distilled. How can I decide? So normally, when your filtrate or when your material in the beaker, this particular conical flask, this is conical flask has become 150 ml. I have taken 20 ml boric acid, and if it becomes 150 ml, then it will be assumed that whatever ammonia was there in the sample has been distilled. Otherwise, what you can do, you can take litmus paper. Litmus paper are in two colors. One is blue color, other is red color. So they can tell you whether the ammonia is still left or not. Because if you use blue color litmus paper, it converts acid into base. Acidic material will, acidic material will be converted from blue to red. The color of change will be red. If you have basic material, then red litmus paper will convert into blue color. And since this ammonia is basic in nature, so if you use red litmus paper, you can take red litmus paper in your fingers, and then put it here. Some drops will come, and if the color remains red, then you may assume that ammonia is not coming. It is already completed. The distillation has been completed. If the color changes from red to blue, that means some ammonia is still left and I can wait. And you can repeat it after 5 minutes to check if any ammonia is left there. So this is how you can decide when to stop the distillation process. Once the material has come, once the distillation is complete, now this ammonia will react with boric acid to form some compound. And then you can take it out. You have one burette. That burette will have some sulfuric acid, 0.9, 0.1 normal sulfuric acid, and you can go for titration. In titration, you will put acid into this drop by drop. This acid is titrant. You will put drop by drop, and then the color will change to red. This bluish green color will change to red or pinkish color. That is your end point. And you can note down the quantity of 0.1 normal sulfuric acid that has been used. Simultaneously, you can run a blank sample. Whatever you have done for the soil sample or plant sample, you need to do the same thing for the blank sample, except that you do not put soil or plant material into the Geldal flask. Then finally or ultimately, you will also get some reading for blank also. So this is used to, calibrate, kind of calibration, because your chemicals may also have some impurities. So therefore, to overcome those kind of errors, we always have one blank sample. Is it clear? This digestion, distillation, and titration. These three steps, if they are not clear, it is time to get clarification. Any question, any clarification from digestion, distillation? Don't get shy. You need to clear your doubt at this point of time. Achappa, any doubt?
No, sir.
Shubhjit?
Sir,
Is it okay or you want to ask anything?
Sir, please explain again the distillation portion.
Pardon?
Distillation portion, sir, एक बार repeat कर देना.
Answer. So in the digestion process, as I discussed in detail, the all kind of nitrogen, organic and inorganic, whatever was there in case of soil or in case of plant, has been converted into ammonium form, ammonical form, and that is basically ammonium sulfate. Now this Jandal flask has nitrogen in the form of ammonium sulfate. And now you add some water, you add about 150 ml water and get it kept for 30 minutes so that the contents are cooled down. After cooling, you can take it to the distillation assembly. The same Jandal flask you can take to distillation assembly. Just a moment. So you put this Jandal flask here, and you need to put some quantity of NaOH, sodium hydroxide, strong alkali. You can put it and immediately close this valve. Immediately you need to close it and attach to the distillation assembly. In the distillation assembly, you have condenser also, which will cool down the hot ammonia. Ammonia will be liberated because of action of NaOH with sodium with ammonium sulfate. It will react with sodium hydroxide, and ammonia will be released. Some water vapor will also be released, and finally here this will be converted into ammonium hydroxide, which will fall into the 4% boric acid mixed indicator solution. Mixed indicator is made from bromocresol green and methyl red. These are the two indicators that makes up this mixed indicator. So, and then you have taken out these contents and titrate against 0.1 normal sulfuric acid. Is it clear?
Yes, sir.
Fine. Now see the reagent. The other part is reagents.
Sir,
Yes, please.
so why we are not adding salicylic acid in plant sample? Salicylic acid and sodium thiosulfate.
Okay. In plant sample, nitrogen is mostly, say, more than 99% or say 100% nitrogen in plant system is in organic form. It is not in inorganic form. Only in few plants like those plants which have very succulent vegetative growth, which are accumulator of nitrates, they may have some traces of nitrate form. Otherwise, our plants on the earth, they have nitrogen in organic form. So because there is no nitrogen in the plant samples, so therefore there is no need to use salicylic acid and sodium thiosulfate, sodium thiosulfate. Because these two chemicals are used to reduce the nitrate, which are present in soil only, They are not present in plant sample. So therefore, we do not need salicylic acid and sodium thiosulfate in plant sample. Is it clear?
Yes, sir. Clear, sir.
Fine. Now we go to the reagents or chemicals that we need. So catalyst mixture, or it is also called as digestion accelerator, as the name suggests. It will accelerate the digestion. It will raise the boiling point. So how to make this in the laboratory? You can take 10 gram of anhydrous sodium sulfate. Anhydrous means there is no water molecule. And if it is hydrous means or hydrated, that means some water molecule is there. For example, ferrous ammonium sulfate or ferrous sulfate heptahydrate, zinc sulfate monohydrate. Monohydrate means 1H2O. But here anhydrous means there is no H2O in the chemical. So take 10 gram anhydrous sodium sulfate and 1 gram pure dry copper sulfate pentahydrate per sample. So per sample, you need to add this. So better to prepare a mixture in advance. You can prepare 1 kg mixture of sodium sulfate and copper sulfate in advance. in this ratio, 10:1 ratio of sodium sulfate and copper sulfate. And you can take total 11 gram. Now, how to prepare mixed indicator? So mixed indicator is used in the estimation of total nitrogen as well as available nitrogen. You will use the same indicator, same procedure. How to prepare it? 0.1 gram bromocresol green, 0.07 gram methyl red indicator in 100 ml of 95% ethyl alcohol. So remember, it is not prepared in water. It is prepared in ethanol. So you need to take 100 ml ethanol, which is 95% concentrated. 95% is the concentration, and put these quantities of bromocresol green and methyl red. Now, boric acid solution, 40 gram H3BO3, that is boric acid, 1 liter of distilled water. Means you take 1 liter distilled water, put 40 gram boric acid and 5 ml of mixed indicator that you prepared, added to each liter of boric acid solution. For each liter boric acid solution, you need to add 5 ml of mixed indicator solution. So your Stokes solution is ready, and out of which you need 20 ml for one sample. Now the color of the boric acid solution along with indicator will be red or pink color. Sodium hydroxide solution is also required for distillation purpose to react with ammonium sulfate to produce ammonia. 40% NaOH solution using nitrogen-free sodium hydroxide flakes. There are some flakes. Flakes means small, like you see your French fries. Khaya hoga apne, you must have used French fries. So they are very small and long. Similarly, you have flakes. Flakes of smaller than that, but shape is like that. So sodium hydroxide flakes should be allowed to stand for 24 to 48 hours. This is very, very exothermic reaction. When you add sodium hydroxide to water, lot of energy is released. And you have to be very, very careful in preparing this solution. You cannot use freshly prepared sodium hydroxide solution. Otherwise, you will be in trouble. It releases very high quantity of heat. So you need to keep the sample of this sodium hydroxide solution for 24 to 48 hours to get it cooled down. You cannot prepare fresh and use it. You need to prepare at least 1 to 2 days in advance. Concentrated sulfuric acid, you also need specific gravity 1.84. Salicylic acid only when you are doing total soil, total nitrogen analysis in soil. Sodium thiosulfate, it is not sulfide. It was slip of tongue. You can correct yourself. It is sodium thiosulfate. I'm sorry. Then 0.1 normal sulfuric acid. And you know how to prepare 0.1 normal sulfuric acid. If you can prepare 1 normal sulfuric acid, then from 1 normal, you can dilute it 10 times. So you can make 0.1 normal sulfuric acid. If you remember, the equivalent weight of sulfuric acid was 49, and you also know the normality, it was 36. So based upon those exercises, I think I need not to repeat. You can prepare 0.1 normal solution of sulfuric acid. Now, procedure, how to do it actually in the lab. Take 5 gram soil sample that has been ground. Ground means powdered and passed through 100 mesh sieve, means 100 holes in 1 linear inch, or simply in 1 length, 1 inch length, how many holes are there or sieves are there. And then for plant, the sample weight is very small, 0.25 to 0.5 gram. Finely ground plant sample to pass 50 mesh sieve in a piece of butter paper. You can take butter paper because the neck of the Jandal flask is very long. If you put soil through the neck, then it can stick to the neck. But if you take the sample of plant or soil in butter paper, and then you wrap it. So it becomes much easier to push the soil or plant sample onto the bottom of the flask. You want everything on the bottom of the flask. So you can use butter paper. Similarly, you can start a blank where you can put only butter paper. No need to put soil or plant sample in that. So here you can see that for soils, normally it is 5 gram sample. For plants, it is 0.25 to 0.5 gram. If you have leguminous seed or leguminous plants, then you can take 0.25. If you have cereal crops, you can take 0.5 gram. Depending upon the concentration of nitrogen, you can increase or decrease the quantity of the soil or plant sample. And now, what you need to do after weighing of the soil or plant sample, you transfer it to 500 ml Geldal flask. Now, then the process for digestion starts. Add 25 ml of concentrated sulfuric acid. Now you add 25 ml concentrated sulfuric acid. Concentrated sulfuric acid, allow to stand, keep it for a few minutes. Then, if nitrate and nitrites are to be included, sulfuric acid added should contain 0.5 gram salicylic acid per 25 ml sulfuric acid. Means you can add salicylic acid into sulfuric acid. You can keep sulfuric acid in a flask, in a beaker, and then you can add salicylic acid into it. Means every 25 ml sulfuric acid should contain 1 gram of salicylic acid. Allow to stand for 30 minutes and then add 5 gram of sodium thiosulfate. Up to this point, you have not started heating it. You just doing it without heating, before heating. So what you have done, 25 ml sulfuric acid has been added to the plant sample. In soil samples, if you are doing nitrate analysis, then you need to add 25 ml sulfuric acid containing 1 gram of salicylic acid. Means this is the ratio of sulfuric acid and salicylic acid, out of which you can take 20 ml. Allow to stand for 30 minutes. Means here, the salicylic acid will play its role. It will react with sulfuric acid and then it will be used to reduce the nitrate. Then you have kept sodium, you need to add 5 gram of sodium thiosulfate. I will explain you the role of sodium thiosulfate and salicylic acid in the reactions. Now again allow to stand for 30 minutes, means after addition of thiosulfate, sodium thiosulfate, 5 gram, you need to allow to stand for 30 minutes. Now you need to add catalyst. Now you are going for heating. Now add catalyst mixture and heat Geldal flask first slowly till frothing continues. Frothing का मतलब है, frothing means is झाग आ जाते हैं ना, झाग. Frothing continues and after that heat it briskly. Then you can put more heat. Continue digestion for half an hour till the digest is clean. Means you can check digest in your hand like this and see if any impurities are there. Any suspended particle should not be there. This should be very clean and transparent. Cool and add 150 ml of water carefully and cool again. Now, the digestion part is over. Now you are going for distillation and titration. Add 120 ml, 40% sodium hydroxide solution along the sides of the Geldal flask and few glass beads. Glass beads are added, means इनको छर्रे कहते हैं, इसके आपके glass के शीशे के छर्रे, glass beads, to avoid bumping. Immediately connect to the distillation bulb. Distillation bulb in distillation unit. Otherwise, if you do not connect them together like this, then the ammonia will be liberated outside. You want it inside the system, inside the distillation assembly, so that the whatever ammonia is liberated should reach the boric acid. So immediately connect to the distillation bulb. Collect ammonia evolved in 25 ml boric acid mixed indicator solution. So you can correct yourself. It is not 20 ml boric acid. It is 25 ml boric acid mixed indicator solution, which is used to collect the ammonia. Distillation should be continued till 150 ml distillate is collected. Titrate ammonia evolved against the standard sulfuric acid, that is 0.1 normal H2SO4. Run a blank simultaneously with a piece of paper, same size as used for wrapping. I already told you, you have to run a blank throughout. Now, to test whether the ammonia has been completely evaporated or distilled, then blue litmus paper to red color if you have acid. If you put acid on blue litmus paper, then the color change will be red. If you have base and put it on red litmus paper, then color change will be blue. Use red litmus paper to check if ammonia is coming. And if no color change means ammonia is completely volatilized. So this is how you use this litmus paper. Now you see the calculations. You have titrated the sample against 0.1 normal sulfuric acid, and you also have a blank. So the amount of nitrogen in the sample, how many grams of nitrogen are there in the soil or plant sample? Formula is ml of acid used for sample, means reading for the sample, minus ml of acid used for the blank, into multiplied by normality of the acid. Normality of the acid was 0.1 normal. If you remember, it was 0.1 normal. So this is the quantity, this is the volume, actual volume of sulfuric acid that has been used. So you can consider it as a V, V1 or V. And normality is N1, V1 into N1, you see. So volume, whatever volume is there, multiplied by 0.1 normal, 0.1 normal, then you will get the milli-equivalents, the milli-equivalents of nitrogen. Then if you multiply milli-equivalents of nitrogen with 14, which is the equivalent weight of the nitrogen is 14, then you get milligrams. So up to 14, you will get milligrams of nitrogen, then multiplied by 10 to the power minus 3. Here means you are converting milligram into gram. So divide by 1000, you will get the grams of nitrogen in the sample by this formula. ml of acid used for sample minus ml of acid used for blank into normality. This is milliequivalent. Multiply by 14, it will give you milligram. And then milligram divided by 1000, you get gram. So many grams of nitrogen is there in your sample. Now you want to convert it into percentage. So percentage, you know how many grams of nitrogen was there, which you have taken as S. So S into 100 divided by weight of the sample. So you get percentage nitrogen in the soil sample or in the plant samples. Normally, total nitrogen is expressed as percentage. So many percentage of total nitrogen in the soil. Hardly we represent total nitrogen in soil with respect to kg per hectare or like that. But we can do so. Now you see the reaction. What is happening under digestion? Are you still awakened or सो तो नहीं गए आप लोग? गुणांक सोए तो नहीं आप लोग? जाग रहे हो? Okay. Now you see the reactions during digestion in soil. You put some chemicals, particularly sulfuric acid in this. You put salicylic acid and you also put sodium thiosulfate. Now what happens for soil samples? This is trapping nitrates with salicylic acid and sodium thiosulfate. These two are used to trap the nitrate, convert them to ammonium. Now you have salicylic acid, and in soil, the nitrogen is present in the form of HNO3. Don't consider it as nitric acid. Of course, it is nitric acid, but it is representing nitrate nitrogen in soil. So now salicylic acid will react with this and it will form nitrosalicylic acid. Now NO2, now nitrogen, or you can say NO3, has become part of this organic compound. Now NO2, this is nitrosalicylic acid. Then this, you also remember the sodium thiosulfate was also there. It will react with the sulfuric acid. It will react with the sulfuric acid and it will form sodium sulfate and thiosulfonic acid. Thiosulfonic acid H2SO3. Nitrosalicylic acid was formed by salicylic acid in soil, which has trapped the nitrates, and it will react with the thiosulfonic acid H2SO3, which was formed by sodium thiosulfate. Now these two, thiosulfonic acid and nitrosalicylic acid, will react in the presence of water to form sulfuric acid is produced, and now this is your aminosalicylic acid. Now nitrogen has been reduced to ammonical form, NH2, ammonium form, you can call it. Then nitrosalicylic acid is further decomposed by sulfuric acid into ammonium sulfate. And CO2 is released, SO2 is released, and water is formed. So these reactions are important. You may try to learn these reactions in this practical. And you can see how intelligently this nitrogen has been converted into ammonium. Now, see reactions, oxidation, digestion of organic material. Now, what kind of reactions were there? How this organic matter, because plants contain some starch, some cellulose, some hemicellulose, that are also to be decomposed. So in organic, whether it is plant sample or soil sample, you have organic matter. So in organic matter, you have organic compounds like glucose, cellulose, starch, whatsoever. It is just a representation of the organic material. Sulfuric acid react and carbon dioxide is released, water is released, sulfur dioxide is released. That means organic matter is broken down. And then trapping ammonical nitrate, ammonical nitrogen. Ammonical nitrogen was there in the sample, soil sample. So ammonical nitrogen can be easily trapped. Ammonical nitrogen can directly react to H2SO4 and it will come into ammonium sulfate. Then your digestion of amino acids or compounds, organic nitrogenous compounds. So R, CO, and H2, these are simple organic nitrogenous compound. Sulfuric acid in the presence of hydrogen, it is converted into ammonium sulfate. So you can see all kind of nitrogen have been converted into ammonium sulfate during the digestion process. And I have also shown you chemical reactions. Now, what are the reactions during distillation? So as you know, after digestion, you have everything in the form of ammonium sulfate. Sodium hydroxide was added and you get ammonia release, water formed and sodium sulfate. So this ammonia is released, it will start liberated, it is liberated and it start moving through the tubes and it will reach the collecting flask of boric acid. Then this ammonia will enter with the water. Water vapors are also there. It will make ammonium hydroxide. So ammonium hydroxide mixed water vapor. These are mixed water vapor. They are moving into the conical flask where boric acid has been kept with mixed indicator. The color of the boric acid was red, but once this ammonia starts entering into the boric acid, the color will become bluish green. So this ammonium hydroxide reacts with boric acid. Boric acid, when it enters the boric acid mixture, and it is converted into ammonium borate or ammonium tetraborate. You can see 4, ammonium borate or ammonium tetraborate. And when you go for titration with sulfuric acid, 0.1 normal sulfuric acid, then this ammonium tetraborate again converted into ammonium sulfate. We have come back to square one, how to say it. We started with ammonium sulfate and come back to again to ammonium sulfate. And boric acid is free. Boric acid is also produced and water. So This is how this total nitrogen estimation is completed in 3 different steps: digestion or oxidation or destruction of organic material, or mineralization of organic material artificially, then distillation and titration. And I have also shown you the calculations, how to calculate the nitrogen in the soil sample or plant sample. Role of nitrogen in plant growth is known to you. CN ratio of material added to soil. I can take one class, full class on CN ratios. Functions of reagents: concentrated sulfuric acid is used for oxidation of soil organic carbon or plant carbon to CO2. Copper sulfate is used as a catalyst or digestion accelerator. It promotes rate of oxidation of organic matter by sulfuric acid. Sodium or potassium sulfate, again raising the boiling point of sulfuric acid, or used to raise the temperature of the digestion. These two accelerate the digestion process. In less time, you can do more. Function of salicylic acid is already explained to you. Nitrates gas lost as nitrogen oxides when soil is heated with sulfuric acid. However, in the presence of sulfuric acid, without heating, you remember we have added salicylic acid before heating. Salicylic acid is nitrated or holds the nitrate. Salicylic acid is used when nitrates are to be included in the Geldar nitrogen analysis only for soil, not for plant. Sodium thiosulfate, same way, reduces the nitrite or nitrate group on salicylic acid to NH2 groups, and nitrate is converted to an organic nitrogen compound and is included in the Geldar nitrogen determination. So nitrate is converted organic compound, and then it is broken down by sulfuric acid to ammonium sulfate. Now, sodium hydroxide, the function, these will be asked in your exam. Functions of different reagents, the principles, and sometimes procedure, sometimes calculations. So whatever I am teaching you would be part of your practical exam. And sodium hydroxide, distillation of ammonia, you have seen, it is used in the distillation process. Boric acid is trapping ammonia coming out on distillation, and 0.1 normal standard sulfuric acid solution for estimating or titrating ammonia trapped in boric acid. Now, dear students, you know about, as I told you in the beginning, that by now you have estimated nitrogen in the soil samples or plant samples. Now we can make use of this information to calculate the crude protein content. As I told you, for estimation of protein in plants or seeds, we estimate only the total nitrogen. Then we can multiply with a factor which may vary from plant to plant, and we can get the crude protein percentage. So it can be calculated by multiplying the total Geldar nitrogen by a factor which vary with the crops. For example, for general factor is 6.25 in many cases like corn, soybean, and in some cases like wheat and barley, 5.7. For rice, it is 5.95, and for sorghum, it may be 5.8. Now, my question to all of you is that why this factor is varying from different crops? Why not the 6.25, just you have 5.7, 5.95, 5.83, and maybe many more variations. Why it is so? Yeah, Astika,
Yes, sir.
please.
So I'm not sure, but the protein content in each of the plant would vary, so the factor is also varying.
Yeah, yes, you have gone to the point, but not the correct answer. But point is related to protein, because proteins in general contain 16% nitrogen. So any protein, see which contains in general, proteins contain 16% nitrogen. So 100 divided by 16, that is why we have 6.25. Means nitrogen percentage into 6.25, we can get the protein percentage. But certain, the protein in other species or other plant or materials, particularly seed, may be different. The content may be different. So therefore, we have different factors. Some may have 14%, some may have 18%. So the percentage nitrogen in protein will vary. Therefore, the factor will vary. So people can determine the nitrogen percentage in the protein, and then factor can be worked out. So this factor has been worked out on their basis. Is it clear?
Yes, sir.
Now we come to the numerical and get ready. Get with your pen, pencil, and paper, calculator or mobile. A 0.2 gram wheat grain sample after digestion. See, the sample weight is 0.2 gram. It was digested with concentrated sulfuric acid and distilled with sodium hydroxide. Simple. Required 3.7 ml of 0.1 normal H2SO4 for titrating the ammonia evolved. Means sample reading was 3.7. A blank was also carried, and which required 0.2 ml of 0.1 normal sulfuric acid. Means sample reading 3.7, blank reading 0.2, sample weight 0.2 gram. Calculate the general nitrogen content in grain sample and estimate the crude protein content in the samples. Because it is wheat, so factor can be 5.7. You can note down these readings, and then I can show you formula, and then you can calculate this crude protein content in the sample. Have you noted this? There are three figures you need to note down. 0.2 gram sample, weight 3.7 ml sample reading, and 0.2 ml blank reading. And then you can go to the formula. This is the formula, and you can calculate.
Sir, 2.45%.
Okay. That is your nitrogen percent or protein percent?
So nitrogen and for crude protein 13.965%.
Any other? So correct, your answer is correct. Dear students, you can see all those who have done or not. You can see the formula is ml of acid used for sample minus blank into normality of acid 14 into 10 to the power minus 3. To put these values 3.7 minus 0.2 into normality 0.1, 14 equivalent weight, 10 to the power minus 3, so that you get value in gram. So your protein in the sample was 0.0049 and sample weight was 0.2 gram. Means in 0.2 gram, sample, you have 0.0049 gram of nitrogen. So percentage, you can convert it in the percentage. So S into 100 divided by sample weight. So you get in percentage 2.45%. And crude protein for wheat factor is 5.7. So you get 13.96. Now come to the next exercise. In this exercise, I will not show you formula in detail, and you start doing it. A 5 gram, that was plant sample, this is soil sample. A 5 gram soil sample digested with concentrated sulfuric acid gave out on distillation ammonia that required 2.2 ml of 0.1 normal H2SO4. Means the sample reading was 2.2. It is just change of language. A blank carried out required 0.2 ml of 0.1 normal H2SO4. Calculate total gel-dal nitrogen in soil. Further, if organic carbon content in soil is 0.56%, this is the carbon, what will be the CN ratio? So you need to calculate the nitrogen percentage, total nitrogen percentage in soil, and then you need to work out the CN ratio. Your time starts now.
Sir,
Yeah,
sir, nitrogen content is 0.056% and CN ratio 1:10.
10 is to 1.
10:1, sorry, 10:1.
Correct. Your answer is correct. Excellent. You are Subhajit? Subhajit?
Yes, sir.
Fantastic. Very good. So these are very basic numericals that students must attempt. And we have one manual, I think I shared. That manual also have some extra questions we have given on this. You can try, or I can make some more questions for you. So now you can see your answers. Sample weight is 5 gram. Reading for sample 2.2, 0.2. So 2.2 minus 0.2, you get into 0.1 is normality, 14 equivalent weight, 10 to the power minus 3 to convert in gram. So your sample contained 0.00828 gram of nitrogen. Sample weight was 5 gram. So therefore, you can convert in percentage, which he correctly calculated as 0.056%. Now carbon percent was given to you, which was 0.6. So 0.6 is to 0.056. 0.56 anupate 0.056. You can get 10:1. Is it clear to each one of you? If anybody has any doubt, it is the time to clarify. Anything up to now, anything, any doubt, if you have, you can discuss with me. Just few more slides are left. Now this numerical also you are required to do yourself. Grain and straw samples of rice in a study contained 1.5% and 0.2% nitrogen. Very simple, you can note it down. Your grain contained 1.5% nitrogen and straw contained 0.2% nitrogen. If the grain yield was 6 ton per hectare and straw yield 6.5 ton per hectare, calculate the total gel-dal nitrogen taken up by the crop. So in this case, you need to convert these grain and straw yields into kilograms, and then you can calculate percentage. For 6, percentage is 1.5, for 6.5, 0.2%. These are the hints. Do it in 1 minute.
Sir, 10,300 kg.
10,300. There may be mistake, some mistake. Just check it.
103.
103. Who said 103? Bolo yaar, dar ke roho, it is correct. 103 may be correct. It's very simple. You may have done some mistake. You can say 6 ton means 6,000 kg. 6,000 kg into 1.5 divided by 100. So it may be 90 kg. And your 6.5 is your 6,500 into 0.2 divided by 100. So if you calculate, it may be 13. Then it is 103. So no problem. We can do mistakes in calculations, but we can correct ourselves. There is no harm in learning. So here you can see grain yield into nitrogen percentage in grain divided by 100. So you get, this is formal, 6,000 into 15.5. So you get 90 kg nitrogen per hectare in straw. And N uptake, grain, N uptake in straw yield into in kg per hectare into nitrogen percentage in straw divided by 100. 6,500, 6.5 into 1,000 into 0.2 divided by 100. So this comes out to be 13. And then total nitrogen removed, grain plus straw. So this is 90 plus 13, it is 103. Is it clear to everyone now?
Yes, sir.
Fine. So डेसिमल में थोड़ा सा कैलकुलेशन में जल्दबाजी की होगी ना आपने, डेसिमल लगाने से हो जाता है. Now do one more. All of you can do it. Grain and straw samples of wheat had 2.2% and 0.4% respectively. If the grain yield was 5 ton and straw yield 5.8 ton, calculate the total gel-down nitrogen taken up by the plant. And second part is also calculate the crude protein yield in grain, straw, and total.
Sir, 133.2.
133.3. Okay, we can.
Yes, sir, nitrogen uptake.
Maybe correct. So you can check the answer. It's not very tough question. Each one of you can do. Some may take 2 minutes, some may take 4 minutes, but I believe that each one of you can do it. If you have problem, you can ask me, I can explain again. It is simple, grain yield into nitrogen percent divided by 100. Grain yield has to be in kg per hectare here. And then similarly for straw yield, you can make the total. And now if you wish to calculate the crude protein content, so you need to calculate the protein percentage. And then likewise, or you can multiply the factor to the nitrogen uptake also. Indirectly you can calculate, or directly also you can calculate by knowing the protein percentage. Protein percentage multiplied by the yields, respective yields, and make the total of grain and straw protein. So now we go to the next. Now you just forget, you can practice it. This PowerPoint is already with you. You can do that numerical yourself. Now last part of this practical is general information about soil nitrogen. Just recapitulating it. Total soil nitrogen content ranges from 0.02 to 0.5% in general in mineral soils. You can correct yourself. The total nitrogen content ranges from 0.02 to 0.5% in mineral soils. In organic soils, total nitrogen content may be more than 2.5%. Total nitrogen content decreases with soil depth, means surface soil have more nitrogen percentage. As you go deeper, the nitrogen content will go down. About 95% of the total nitrogen in surface soil is organic nitrogen, that is already discussed. I gave you range 95 to 99%. The stable soil organic matter contains about 5% nitrogen. You remember, stable organic matter means humus. Humus contains about 5% nitrogen and 50 to 58% carbon. 50 to 58% carbon, and CN ratio is 10 to 12 is to 1. In soils, generally the CN ratio is 10 to 12 is to 1. Generally, nitrogen mineralization occurs with residue nitrogen content and more than 2% under aerobic conditions. So on an average, 2% is the accepted value. 2% is the accepted value. Now, general information about soil nitrogen. If nitrogen content in surface soil 0 to 15 centimeter is 0.1%. See it critically, see it more carefully. If nitrogen content in surface soil 0 to 15 centimeters, somebody is giving you 0.1%. Then how can I calculate how much nitrogen is there in quantitative terms? So then total nitrogen kg per hectare will be, you can take the soil weight 2 into 10 to the power 6 into 0.1%, like you did in case of, just now you have done in case of grain and mixture uptake. So here also you straightway multiplied by 0.1%. 0.1% means 0.1 divided by 100. So 2 into 10 to the power 6 into 0.1 divided by 100, you get 2000 kg per hectare. So if you are given the soil weight, you can use. If soil weight is not given, you can always take 2 into 10 to the power 6 as weight of the 0 to 15 centimeter soil, and multiplied by the percentage, straightway you will get the quantity of nitrogen in terms of kg per hectare. Now, inorganic nitrogen, that is ammonium, nitrite, and nitrate, represent 2 to 5% of the total nitrogen. If total nitrogen in soil is 2000 kg, now we are taking the same example forward. If the total nitrogen is 2000 kg per hectare and inorganic nitrogen 4% of the total nitrogen. Here it is not 2% because range may be from 2 to 4%. So if it is 4% of the total nitrogen is your inorganic nitrogen, then inorganic or mineral nitrogen is equal to 2000 into 4 divided by 100. Then you got 80 kg per hectare of ammonium plus nitrate nitrogen. Then the available nitrogen. Now question comes, available nitrogen. This is mineral nitrogen 80 kg. Now we see the available nitrogen. For available nitrogen, you need potentially available nitrogen or readily oxidizable nitrogen. Here, mineral nitrogen that you worked out 80 plus readily oxidizable from organic nitrogen. So if you add these two, means mineral nitrogen plus readily available, readily oxidizable, then it becomes your available nitrogen. Generally 1 to 3% of total nitrogen to be potentially released from tropical and humid subtropical soils in a year. So these already discussed so many times. Now, just consider it can be 2% release, it can be 1%, it can be 3%. Consider 3% release rate per year. So 2000 into 3 divided by 100 is equal to 60. So 60 kg will be potentially mineralizable nitrogen. So thus available nitrogen will be 80 kg plus 60 kg is equal to 140 kg. This is how we can calculate the available nitrogen. You see how we have calculated mineral nitrogen. We assume 4% of the total nitrogen is mineral nitrogen, or it can be 2%, it can be 3%. And then 1 to 3% of the total nitrogen to be potential. So you need to remember these values. Now, here is a question for you. Calculation of nitrogen mineralization. If anybody misses anything here, what I suggest you can read, take help from the Tisdale book, Soil Fertility and Fertilizers. You already have that book. You can find 2-3 pages devoted to this aspect. I have learned from there. So if you miss any point, if anything is not clear, I am trying to make everything clear to you. But still, if you have any doubt left, you can see those pages. Now, calculation of nitrogen mineralization, kg per hectare nitrogen mineralized, 15 centimeter depth. This is the formula given to you. This is weight of the soil kg per hectare. How to get it? Total nitrogen mineralized. Weight of soil kg per hectare into percentage soil or organic matter. How much percentage of organic matter is there in my soil? Then what percentage of nitrogen in soil organic matter is present? What is the percentage of nitrogen in soil organic matter multiplied by percentage of soil organic matter mineralized? That is readily oxidizable. So these are 1, 2, 3, 4 things. 4 things you can multiply each other and you will get the kg per hectare nitrogen mineralized. First thing is weight of the soil. Second is percentage soil organic matter, means organic matter in soil. Third is percentage nitrogen in soil organic matter. And then percentage of soil organic matter that can be mineralized to give nitrogen. All are in percentage except weight of soil. So weight of soil is 2.2 into 10 to the power 6. Percentage soil organic matter range from 0 to more than 75% in histojoules. Soils and values between 0.5 to 5% are most common. So value may be given to you that so much is the soil organic matter percentage. And then the amount of nitrogen in soil organic matter, here it is assumed as 5%. 5% nitrogen is there in the soil organic matter. And percentage soil organic matter mineralized in 1 year, it depends upon texture, climate, management. But on an average, 2% in fine texture soil and 3.5% for coarse texture soil. So in coarse texture soil, the decomposition rate is faster. So here 3.5% may be broken down, and higher values in warm climates, lower values in cold climates. So these values vary, but generally average is 2%. Now you can solve this problem. Calculate the amount of nitrogen that could potentially be mineralized in soil on the basis of following information. Weight of soil 2 into 10 to the power 6. Number 2 is percentage soil organic matter. Here 2.5% soil organic matter is given. The amount of nitrogen in soil organic matter, it is 5%. Percentage soil organic matter to be potentially mineralized in 1 year. Instead of 2%, you are given with 2.4%. Now you can put all the 4 things into the formula and get the answer. So kg per hectare nitrogen mineralized 15 centimeter depth is equal to weight of soil into percentage soil organic matter, percentage nitrogen in soil organic matter, percentage soil organic matter mineralized. So you put these values and you get 60 kg per hectare. So 60 kg nitrogen can be provided, can be released by the organic matter in 1 year. So this is potentially mineralizable nitrogen or readily oxidizable nitrogen. You can see how you can calculate it. The last slide is calculate the amount of nitrogen that could potentially be mineralized in soil 15 centimeter weight on deep on the basis of following information. So you given soil weight, percentage soil organic matter 2%, amount of nitrogen in soil organic matter 4.8, percentage soil organic matter potentially to be potentially mineralized is 2.6. Now can you do it? Please do it. Anybody? You need to multiply 4 figures, gentlemen. 2.2 into 10 to the power 6 into 2 into 4.8 into 2.6, and these may be divided by 10 to the power 6. So 10 to the power 6 से 10 to the power 6 कट गया. So 2 into 2, 4, 20, 40 के आसपास आएगा. It will come around 40, 45.
Sir, 49.92 kg per
Okay.
hectare.
Good. So now this practical is over, and those who have not seen Dr. Rajendra Prasad can see him here. Have you heard his name? Dr. Rajendra Prasad?
Yes, sir.
Yeah, he was our teacher and our guru. He is now 88 years old, and he is on the bed, unfortunately. So we have a break for 15 minutes, and I have you can open your videos for a few minutes, few seconds, sir, madam. Hello. Yeah. So did you enjoy this practical, or it was just you were could you learn anything from this practical? It is theoretical practical, you can say. कुणाल क्या हुआ फोटो को आपके? बोलो भाई, ये अमर हो जाएगा वीडियो. ये मैं डाल दूंगा यूट्यूब पे 2-4 दिन बाद. सो ये पता नहीं कब तक रहेगा. आपका भी फोटो आ जाएगा वीडियो. कंचन, so any point, anything you want to clarify in this practical, it is really good time, right time to ask. I can clarify all your doubts in this practical.
Sir,
Yes.
sir, if we are getting the ammonium sulfate in the first step only, then why we are going for distillation and titration?
Actually,
Because ultimately at the last also we are getting ammonium sulfate only.
yeah. Actually, ammonium sulfate is directly produced in case when we have ammonium on. If we have ammonium, all nitrogen in the ammonical form, then we need not to go for any other thing. We can straightway go for distillation. But your nitrogen is not in available ammonium form. Some is in nitrate, other is in organic form. So those are to be converted into ammonical form. And it is that by chance that we have used sulfuric acid, and which react with your ammonium, and it converts to ammonium sulfate. So we need to add sulfuric acid, not just to form ammonium sulfate with mineral ammonium, but to decompose the organic matter. I don't know whether I could answer or not. Or you can
Yes,
ask
sir.
your question again. You can ask your question again.
Yes, sir, clear.
Clear? Okay. So अच्छा आपको समझ में आ गया practical? You got It clear?
Yes, sir.
So we meet after, yeah, any other question? No problem. We meet after, I will have just your 20 minutes more, not more than that, because available nitrogen practical by Subaiya and Asija, 1954, is very simple method and very close to this method. So unnecessarily, I will be taking at some other time. So now it is better to take that practical also, only for 10, 15 minutes, 20 minutes. So we meet after at around 5. Is it fine? Or 5 past 5? Okay, 5:05 पे मिलते हैं. Okay?
Yes, sir.
या क्लास है कोई आप? We meet at 5 past 5. Thank you.